Hello and welcome to CEM and Lucidity Systems Wednesday webinars on separation solutions. Today's presentation, utilizing compact integrated chromatographic instrumentation for pharmaceutical analysis, is presented by Dr. James Grinyas of Rowan University. Before we get started, let's review some of the features of GoToWebinar. The audio for this presentation is available on your computer only. If you cannot hear the audio, please check your speaker volume. To access the handouts for this presentation, click on the handout icon and then click the handout you want to access. The handout file will automatically start downloading from your default browser. At the end of the presentation, we will have a question and answer session. You are welcome to submit questions at any time during the presentation. To do this, please type in the question section and click Send to submit your question. Please note that not all questions may be answered live, but they will be answered by email after the webinar concludes. It is now my pleasure to introduce today's presenter. Dr. James Grinyas is currently an associate professor in the Department of Chemistry and Biochemistry at Rowan University in Glassboro, New Jersey. His research interests include improving the throughput and efficiency of chromatographic separation and the miniaturization of chemical measurement techniques. He received his PhD from the University of North Carolina at Chapel Hill in 2014 and then moved on to a postdoctoral fellowship at the University of Michigan until the end of 2016. James has received a number of awards for his work to date, including the HPLC 2013 Csaba Horvath Award, the 2020 Young Investigator Award from the Chinese American Chromatography Association, a National Science Foundation Career Grant, the 2021 American Chemical Society Young Investigator in Separation Science Award, and the 2022 LCGC Emerging Leader in Chromatography Award. He was also named the Analytical Scientists Top 40 Under 40 Power List in 2018. To date, he has published over 30 articles and given over 80 oral and poster presentations. With that, we are ready to begin our presentation. Jim, over to you.
Thank you all for coming today. Today we're going to be talking about utilizing a compact integrated chromatographic instrument, specifically the Lucidity miniLC system for pharmaceutical analysis and primarily for over-the-counter analgesic drugs based on published USP-style monograph methods. I'd like to thank my student Alexis Zimmer from the Grinyas Lab at Rowan University, who helped put together a lot of the characterization of the compounds that we're going to talk about here today. So here in the United States, we have this United States Pharmacopeia National Formulary compendia of analytical methods or monographs for testing drug content, purity, and other specifications. And there are similar pharmacopeial methods and compendia that are available for a number of different jurisdictions as well. And amongst all of these sets, EP, JP, CP, USP, and many others, many of the monographs that are listed in these collections require LC analysis for both the active pharmaceutical ingredient and any impurities that may exist in the drug formulation. And as you can see here, just a sample of what some of these monographs look like, one for ibuprofen, one for naproxen and naproxen oral solution. And it details where to get, supplies for doing this method testing, method conditions, and a variety of other factors that an analytical chemist would need to help perform the analysis. Something we've been working on here at Rowan University is, as part of some of our research-based courses, students who want to learn a bit more about performing LC analysis, especially for pharmaceutical compounds, as we have some graduate programs that are related to pharmaceutical chemistry. We've been trying to teach them some of the basic skills that they'll need in today's modern pharmaceutical industry, and the miniLC has turned out to be a great tool to do that in terms of its intuitiveness and ability to do some method development, method translation, and eventually some method validation and qualification. And as you can see here, what we've done in this example is using the miniLC, taken the published method for naproxen oral suspension and translated to the miniLC platform. Basically, this is the assay you would do to ensure the content of the active pharmaceutical ingredient naproxen for some sort of over-the-counter naproxen oral solution, and the quantification is performed through an area ratio using ethyl paraben as an internal standard. As you can see, great resolution between the two compounds for this assay. But with the way that many of the monographs are published, the methods can be somewhat slow. And so there's been going on nearly 20 years at this point, a big initiative for monograph modernization. And there are a lot of monographs that rely on outdated non-specific techniques, whether it's titrations, spectroscopy. There are even occasionally some ones that rely upon flame test. And so having more chromatographic based methods, especially LC, but expanding it beyond, maybe single wavelength detection to go to photodiode array detection, something like a universal detector like a refractive index or charged aerosol detector, and even mass spectrometry a bit. Try to do some gradient methods, and create general methods can be applied to lots of different active pharmaceutical ingredients. And over the past 8 years, over-the-counter monographs have had a special focus because there's a goal to make sure that specificity and sensitivity requirements are met for a variety of products, as well as impurities and excipients that exist in those drugs. And this is especially important because for a lot of these over-the-counter analgesic drugs, they're manufactured all across the world. And so you think about the different manufacturers who might be working on these compounds, making sure that these methods translate well from lab to lab is an important technique. And so in terms of things you might do, not only do we think about trying to make the methods go faster, but perhaps be a bit more robust as well. And when we talk about chromatographic methods within the USP, there's chapter 621 that gives a variety of guidelines that allow what types of allowable adjustments can be made to a given method. And some of these things can actually be applied to help increase the throughput of your analysis, which can help, identify any issues that might exist during the manufacturing process and try and remedy those as fast as possible. And as you can see here, there's a number of things that relate to both the mobile phase preparation for a method, the column selection for a method, various different instrument parameters. And so what I'm going to do today is talk a little bit about how some of these allowable changes may factor into a translation to a different platform, and specifically highlight some of the ways that you can investigate these changes using the Lucidity miniLC platform. And so one of the first listings is adjustments that can be made for the pH of a mobile phase. And so you can see here for the ibuprofen monograph, the way that the mobile phase preparation is listed in the actual monograph. And then you can talk about how adjustments can be made under these chapter 621 system suitability guidelines and allowable changes. And so what's listed is now, and I will note that in the near future, there are probably going to be a number of changes that are published for some of these system suitability criteria, and there will be variations in terms of what is and is not allowed, especially with some shifts to more gradient-based LC methods. So this is under some of the existing guidelines now, but pay attention to some of the upcoming releases from USP in the near future to see if there may be some further updates beyond what we talk about here today. But for this specific one, I think the biggest question you have to really ask is what compounds are you looking at, and specifically, are they neutral, or do they have some acidic and basic groups that could change if you change the pH? And I think the biggest thing is how close is the pH that your mobile phase is being prepared to the compound pKa, because if you are close, you could see some effects on retention and potentially even peak splitting, where you get a weird peak shape if you're very close to that point and you're getting a mixture of a protonated and a deprotonated compound. So this is something you need to be careful of depending on what types of analytes you're looking at in a specific method. Also, in terms of buffer concentration, then not only do we have to worry about what the pH is, but what's the concentration of the buffer that we're going to add. We want to make sure that we have sufficient buffer capacity that if we load up a higher amount of compound, that's probably most typically going to be seen in the drug assay testing, where you're focused a bit on what's the actual concentration of a given formulation of some over-the-counter analgesic drug. Those concentrations are going to be much higher than what you would expect for an impurity. But it also depends on what type of chromatography is being performed. So many published monographs call for columns in the L1 category. We'll talk a little bit later today about the various column categories. But C18, falls broadly under L1, and the buffer concentration itself probably isn't going to have too big of an effect for most reverse phase separations, especially if we're talking about, separation of these neutral compounds. However, for L3 classification separations, which here are bare silica, but so it would operate under a, generally a Hillock type separation. There you might see some kind of change because buffer concentration can play a big role in the way a Hillock separation is performed and what effects we see on retention. So that is one thing to watch for, is what column is it calling for in terms of this mobile phase preparation method adjustment that's allowable. In terms of the composition, okay, this is where things can get a little bit trickier because you are looking at, in terms of the allowable change, 30% relative or 10% absolute. And so it will depend on what the published monograph calls for. And again, a lot of these methods are isocratic today, although there may be some changes into what will be allowable, in terms of gradient shifts. This one, you could see a significant change for some compounds in reverse phase separations or an L1 column designation, C18. There are some other reverse phase categories for things like C8 as well, but L1 is the big one. And so, with a C18 column, it's possible that with a 10% change in your organic component, you could see a 2 to fold change in retention factor and sometimes more. And so in some ways, if you are able to increase the organic content within this allowable change, you might be able to increase the throughput. But in terms of system suitability criteria, especially what's published in a specific monograph, oftentimes there is a set level of resolution that needs to be obtained between, an active drug compound and the internal standard, and the impurity, depending on what type of testing you're doing. And so you want to make sure with these changes that you're still meeting your system suitability criteria. We'll talk a little bit later as well about robustness, but one thing might be not necessarily that you're planning on making this change, but you want to have a broad range of what is acceptable in terms of the overall preparation criteria. How robust is your method to, a change of plus or minus 10% within the method. And again, it's 30% relative or 10% absolute. So it really depends on if you have a very minor component in your mobile phase. There are some that are on the order of 10 to 15%. Their 30% relative may not allow for a 10% full change. You may be limited in terms of how much you can shift that amount. So in terms of setting up these methods in the miniLC software, on this method development page, you can see here this is a separation of ibuprofen and valerifanone for its assay. And in that case, what we're doing is being able to change the composition, potentially being able to create an isocratic or a gradient method. Again, here we're typically doing isocratic for most of these monographs before these next set of changes are going. But one thing you may also want to consider is, do you want the mixing to occur online, or do you want to pre-mix the mobile phase? And one way to sort of look for differences in those two cases is what your baseline noise looks like. Are you getting sufficient mixing? And typically there is sufficient mixing. That's not too much of an issue with modern LC instrumentation, but still something to keep an eye for. And if you were to do a pre-mix, in that case, you would then generally just have 100% of either your A or B mobile phase line. The next adjustment is detector wavelength, and generally you're not supposed to change it, although there can be small errors. And this isn't really a problem in major modern instrumentation, including this miniLC that we're talking about today. But I think the bigger thing is maybe understanding, from a fundamental level, why are certain wavelengths selected for a given monograph. You can see here for the ibuprofen monograph, again, one of the main examples we're using in this talk here today. Generally 254 nanometers is selected, and if you look at the concentrations that are prepared, the concentration of the active pharmaceutical ingredient API, the drug assay for ibuprofen, that concentration is much, much higher than the expected concentration we're going to see for the impurity. And that's to be expected, right? You hope that your impurity is going to be in a minor component relative to your active pharmaceutical ingredient. And generally the selection is made in such a way that you're maximizing the signal of the impurity peak, so you have good detection limits for that compound that's going to be in a trace amount, but that you're still able to easily see the drug product. And I think in a lot of cases there, the concentrations have been selected to give you reasonable detection limits for that compound as well. And so you can see here, looking at these two absorbance spectra for both ibuprofen and ibuprofen-related compound C, which is the primary impurity that you might be looking for in this monograph assay. 254 nanometers gives you a huge signal for the impurity compound, and so you'll be able to even see it at smaller amounts, more than you will of any other typical wavelength that we might use for a LC-UV detection, you know, maybe ranging from 210 nanometers up to 280 nanometers. And so just to kind of give an example of that, using the miniLC, if we go in here, this is where you actually go in to set the detector wavelength, and from there, what you'll end up doing is running your method once you go through and put this in the system sequence, which we'll again talk about in a minute. And so you can see here we ran the same impurity compound, this compound C, through the column at 3 different wavelengths: 220 nanometers, 254, and 280. At 220, you can barely see a peak. You have to kind of squint to be able to go in and see that little bump above the baseline. This is all on the same y-axis scale. At 254, the maximum wavelength, you can see clearly the compound C peak is there, and then at 280, again, it gets smaller. So again, this is generally how the monographs were developed when they were originally published to maximize these signals, and so that's something you don't really want to vary. You want to make sure that you set that to the proper setting when you go in and do your method development. Now, in terms of injection volume, generally there's instructions in terms of what injection volume you're supposed to use, as well as then how many replicate injections you do. That's typically more in terms of the system suitability criteria and doing more of the qualification and validation of the method. But we're going to talk over the next few slides about what column changes can you make. And so there are going to be some shifts we're going to talk about in terms of flow rate to accommodate changes in column diameter, and oftentimes injection volume is also scaled when you change column diameters like you will with flow rate. And so what you're thinking about in these cases is there's going to be some kind of trade-off between detection limit, because you want to inject as much volume as you need to be able to properly view your impurity peak, which again is going to be your trace component. But at the same time, for a given column volume, if you inject too much, you could see overloading. But more typically with modern columns, the bigger issue you may end up seeing is excessive band broadening, especially for these isocratic compounds, where in a gradient method you might get some peak focusing at the head of the column due to changes in the mobile phase strength across the gradient, where as an isocratic method you're not going to see that nearly as much, especially for poorly retained compounds. In terms of column temperature, again, generally there's a prescribed temperature at which the system is about to run. That's another thing that you can set inside the method that you run within the miniLC software. In terms of what you change between plus and minus 10 degrees C, generally in reverse phase separations, you're going to see a decrease in retention time as you increase the temperature. So again, that might be another way, along with increasing the organic component of your mobile phase, to try and speed up your method a little bit, increase the throughput. But again, remember that the resolution between our components must be maintained to whatever system suitability criteria is listed for a given method. And so you can see here, column temperature, injection volume, as well as needle washes to prevent carryover from run to run. These are all things that we can go ahead and set up in the miniLC software in terms of this method parameter page. Now, the biggest shifts, and this is something where, again, there's going to be more updated guidelines, again, with the impending changes that may be coming to Chapter 621. But as published right now, in terms of the allowable adjustments for column length and particle size, typically what you're thinking about is either a ratio of length to particle diameter, or in general maintaining efficiency with some kind of separation. So again, very frequently for these older monographs, you see somewhere between a 3.9 and 4.6 millimeter ID column, generally 15 to 25 centimeters in length. And we know that in modern chromatographs, shifting down to a 3 or even a 2.1 millimeter ID column, is generally feasible. And so it depends on how much of a change you want to make. In terms of method translation, keeping the column dimensions the same is the easiest and maybe most applicable if you're doing a direct change. But if you want to, do some updated changes, you can shift here. But the things you need to think about are what column dimensions are available, and if I run those column dimensions, especially if you maybe move to a smaller diameter, are you going to be able to maintain that efficiency as well, because you may have differences in terms of the system contributions to broadening as the column volume goes down. So within chapter 621, there is this table that's given that gives, a list of some of the acceptable adjustments that you might be able to make in terms of some of these changes. And what it does is for a given column, in this case 4.6 by 150 millimeter with a 5 micron packing, it kind of uses that as the length to particle diameter ratio of 30,000. So you're thinking about, 150 millimeters divided by 5 micron, or 0.005 millimeters, and so in that case you end up getting this 30,000 ratio. But that case they're listing that as okay, you're going to be whatever flow rate you're listed, so maybe that's 1 mil per minute or 2 mils per minute. There's some given flow rate and a set efficiency with that and a set pressure, as well as a rating of let's say 1, normalized time of 1 for the runtime. And so then you can see in terms of those ratios, then for all these other dimensions, how should you scale the flow, how should you scale the runtime, and what kind of changes to the pressure and the efficiency might you expect as well. And then going down to 5 centimeter or 50 millimeter length columns with both 4.6 and 2.1 millimeter IDs, and then a range of particle diameters, going from 5, some of the older column technology, down to sub 2 micron particles that are a bit more common now. But you can see in those cases that if our allowable change is related to length to particle diameter ratio or the efficiency, plate count N, you can see that 30,000 number, dips to as low as 28,600 as we go to these other dimensions, but remains effectively, nearly constant, and that efficiency N is 1 down all these columns as well. So, these kind of give guidelines of how you might scale the flow rate and what change you would then expect in terms of runtime. And we're going, as we go to the 4.6 or 2.1 millimeter ID by 50 millimeter length with sub 2 micron particles, we're maintaining that efficiency and ratio of the length to particle diameter, but we were able to potentially run things 10 times as fast. Okay, our runtime going from 1 to 0.1. So that might be, one thing. But again, what sacrifice do we make? The pressure is nearly 9 times higher. So these are all things you need to think about as you do this method development. Now, this is an example where here we made a shift from 4.6 by 250 millimeter columns with 5 micron fully porous particles down to 4.6 by 100 millimeter column with 2.7 micron superficially porous particles. So this is a slight shift because there are, a lack of guidelines right now. Again, this is going to be one of the things that gets changed in the upcoming reports in terms of fully porous and core shell or superficially porous materials. That's been something that's more recent change since 621 was originally published. But in these cases, what we were able to do is make the method about 2 to 3 times faster, even though we only had to make a slight shift in the flow rate. So you can see here, this method generally takes on the order of 7 or 8 minutes. We were able to get all the peaks to elute in about 2.5 minutes in this case. And again, this was performed by translating the conditions that were given, changing the column type, but then really just putting the method parameters right into the miniLC method parameter software. Now, when we talk about robustness, and we're going to talk about that again in a little bit, usually these columns just say C18 or L1 column, but how many, columns fit under the L1 USP designation? You can see here, hundreds of columns listed that fit under there. And one question you might want to think as you do this method development or potentially translate the method from one system to another, one location to another, is how similar are these columns? And so the USP has published the PQRI approach that allows you to select columns for equivalent selectivity. And really what this does is takes a database of existing C18 columns, existing C18 phases that have been characterized using the hydrophobic subtraction model. And it's a little bit beyond the scope of what we're going to talk about today, but effectively there are a set of analytes you can run under a set of a couple different mobile phase conditions that will allow you, based on the observed retention times, to back calculate out a series of effectively coefficients that correspond to various fundamental characteristics of reverse phase retention, and based on those coefficients, then you can get a good estimate of how close two phases might be to each other. So even C18s are not created equally. There can be a lot of changes in those C18s based on the base silica material, end capping, all sorts of things can vary that process. And so this allows you to at least help select what columns may or may not be, similar for a given method. And so yeah, if you just search USP PQRI, that'll get you to the database that allows you to do this. And there are a couple other databases that exist online as well that allow you to make these kind of comparisons. Now, when we think about all these different columns, we saw that there was potentially changes that could be made in terms of the column diameter, and that's where you need to start thinking about the flow rate change that exists. So if you make a change in column diameter, that's when you're probably going to make some kind of shift in the flow rate, and what you're trying to do is make a change for keeping the linear velocity constant. So there's a general formula that can usually be considered where you take the, if you keep the packing material the same, really you're just worried about the shift in column diameter. It's really just looking at the square of the ratio and scaling the flow rate appropriately. So if you decrease the diameter, you're going to scale the flow rate down accordingly. And generally you might be doing the same thing with the injection volume scaling, but again, the big thing with the injection volume scaling is to make sure that you are still meeting, some kind of detection limit system suitability criteria. So again, as we think about setting up the methods, so you can set the flow rate in your actual method parameter setup, but then also, at any given time in the miniLC software, you can easily see what the flow rates are for your A and B pumps to give you a total flow rate, as well as the overall system pressure and the temperature that the column oven is set at. Now, when we think about qualification eventually down the line to validation, robustness parameters and various qualification criteria come into play, and this relates to mobile phase composition. So again, within those allowable changes in some given range, how reliable or how repeatable are your retention times, if there is a small change within that allowable list for pH or the percent of organic component. Same with the column oven temperature. Same with how close is one L1 column to another column. So you might test from a few multiple sources. And again, when you make some kind of set validate method, you might say this is the preferred column, but these other columns potentially are allowable as well. And then are there slight changes that might be allowable in terms of flow rate or injection volume? If you don't set it exactly, are you going to get the same results out? And the things that we think about in terms of the suitability for these techniques is, does the method remain linear? Is it repeatable from run to run in terms of the calculated quantities for a peak? So usually that's a peak area ratio to the internal standard. That relates as well to the recovery, where you might be making a calculation from the linear calibration curve that you generate. And then as well, are your peaks still resolved sufficiently to make sure that you don't have any sort of coelution or overlap between them. And linearity ranges typically dip down to on the order of 0.01 or 0.05% of your active pharmaceutical ingredient content for a purity that you're looking at. And in terms of the active pharmaceutical ingredient itself, especially for these OTC drugs, generally you're looking at about 75% to 125% of what the expected API content level should be. And so as we look here, how do we rate these criteria? So linearity, a 5+ point calibration curve when we're looking at the R² value across those ranges that I just mentioned. Repeatability, what's your percent RSD if you do replicate injections? Recovery, again, we're going to relate to that peak signal, and then a calculated value from the calibration curve, and then your resolution, again, generally between the analyte, whether it's the API or the impurity and the internal standard. We also want to think about intermediate precision, which some people kind of falls under the broad category, something called ruggedness. And so day to day, how do these things hold up? What about running, different instruments, so different instrument vendors in the same lab? What about running the same instrument but in different labs, or different people running the same instrument? And so you can imagine, you have your miniLC in one lab location to do method development and, R&D type things, but then you have a second one for quality assurance, quality control monitoring at some manufacturing plant somewhere else, right? So if you put the miniLC in both those spots, you have different people running them, preparing the calibrations. Are you able to maintain that method consistency from place to place? And then again, that interlaboratory reproducibility. So these are all things we need to think about under this intermediate precision or ruggedness. How solid is it? And that becomes especially important as we think about method translation to, again, manufacturing type sites, which is common in these OTC analgesic type methods. And so when we think about ways to automate this process and simplify it a little bit, the miniLC comes with an integrated auto sampler where you can configure the run, so you can prepare all your different concentrations for your various calibration curves, set it up to do multiple injections from those samples to make sure that you're getting the proper number of points to generate your calibration curves, or do replicate injections for a given percent within your percent API value. You can see here they're kind of arranged in this circular auto sampler format. So you tell it the vials where to go. In the software, you configure it to say, okay, inject this many times from this vial spot, inject this many times from this vial spot, and then you'll be able to sort of just let it go from there. And then more recently, the software has been updated to allow this calibration feature. So you can actually tell it what concentrations you're expecting for these compounds, which concentration is listed in each of these vials, and it'll actually generate that linear calibration curve for you. You can see this example that they did at Lucidity headquarters to be able to do this for caffeine as an example study. And so with that, just to kind of conclude, what we've talked about today is a bit in terms of method development and eventually transfer that can be done for pharmacopeial monographs, especially OTC analgesic drugs, and the guidelines for allowable changes throughout Chapter 621 for these existing methods. And many of the published monographs rely upon HPLC methods that should be readily translated to the miniLC platform. We demonstrated that for ibuprofen and naproxen here. We're in the process of exploring aspirin and a few other compounds as well. Both, sometimes daytime mixtures that include caffeine, various things like that, and demonstrating those capabilities on this integrated system platform. And with the Lucidity software, there are a lot of protocols that allow the ability to automate some of the tasks that you need to do through the use of the auto sampler and some of these calibration features. With that, I'd like to thank you for attending, and let me know if you have any questions.
Thank you, Jim, for sharing the exciting work that you and your team are doing. We will now begin our question and answer session. As a reminder, to submit a question, please type it in the space provided and click Send to submit.
Okay, and joining us for the question and answer session today is Dr. Alicia Stell with CEM and Lucidity. So I'll just go ahead and dive right into our first question. So the first question is, how might the method transfer process be different for gradient methods?
Great, thank you for the question. So a lot of what we talked about today in terms of these allowable or acceptable changes focused on isocratic methods. And I think you maybe saw, if you look at some of the slides that mention mobile phase, that changes are acceptable for isocratic methods, but it usually says no or in part for gradient methods. I will note that there are some changes coming to the USP, especially Chapter 621, that relate to how you can adapt and transfer gradient methods. So there will be some more coming out that from the regulatory side sometime soon. But in terms of thinking about just a general method transfer process, I think the big thing going from one system to another is, what is the gradient delay volume, and how might that shift if you just transfer method directly? How long does it take for that initial change in the mobile phase condition to reach the head of the column? And so there's ways that you can test that by adding a small tracer. Typically people will use something like caffeine in their organic component. I guess I'm referring primarily to reverse phase. And so you can look for when that baseline shift happens. And so once you make that assessment change, you can do some calculations. I won't go into the full theory here. And if anyone ever is working on that and translating the method of the miniLC, I'm happy to help them with that process. But effectively what you're going to do is do that test to figure out what the delay volume is, and then make some changes to your gradient program to account for that.
Okay, thank you. The next question is, have you used this system for other applications outside of pharmaceutical analysis?
Oh, great question. Yeah. So today, with the time we had, I figured to primarily focus a bit on pharmaceutical separations, but we have used the system for a few other applications that we're interested in. So we do have some interest in using small systems for the separation of drugs of abuse in various clinical samples. So we have done some testing on mixtures of benzodiazepine drugs using the miniLC. And then we've done a few other things here or there. We did do a quick test for capsaicin. And then we've also done some other pharmaceutical separations that I didn't talk about here today, in this same sort of uh, monograph type realm. We've also done a lot of fundamental work using a set of standard alkyl phenols that people use for general performance testing and column testing. I know that there are a lot of other application notes that are available on the Lucidity miniLC website, so maybe I'll turn it over to Alicia briefly to mention some of the other applications that they've been working on down at Lucidity.
Yeah, thank you, Jim. Yeah, for those listening, you can go to luciditysystems.com and go to the application page and see kind of the stuff that we're working on. But just to kind of give an overall summary of it as far as what we're seeing our customers, you heard Jim talk today from pharmaceutical applications within a university setting. So academia is definitely where there's a lot of benefit to smaller system, but also what we're finding is people that would typically maybe not consider the fact that you could use something like a miniLC in your lab, that they're probably maybe outsourcing that data because it was, maybe too complicated or just not possible at the time to take this system, to have a lab within your environment. But outsourcing data is becoming a lot more expensive and harder as far as timing is concerned. And so there's a lot of people considering, can I do this in my lab? I think you'll find that within Lucidity, with these products, it really opens up that possibility. So the applications are kind of endless as long as you're using a UV for a miniLC, a LC UV system. We also have a mini GC which is a GC FID system, which offers the same benefits.
All right, thank you, Jim.
Yeah,
and Alicia.
actually, maybe before we move on to the next question, Alicia's answer did stimulate my memory about one thing I will note as well. So, about a year and a half ago, our university, our own university, launched an institute for cannabis workforce development and research. And so, a lot of companies in the state of New Jersey are starting, cultivation and testing facilities. And a number have toured our facilities, and in my lab particularly, we have larger standard modular benchtop LCs as well as this small integrated miniLC. And a lot of these companies are small startups who don't have a lot of laboratory space, and they need to maximize what they have as much as possible. And I think that they talk to a lot of the companies that make these bigger LC systems, and they think, okay, this is what my potential benchtop footprint is going to be, and then they see the miniLC and how little space it takes up, and their eyes kind of bulge out and they get excited. So I do think that especially as as Alicia mentioned, people take these testing needs that they have in-house, the small integrated system definitely provides an advantage there.
Very good, thank you. Let's see, the next question is, what considerations should be made for column selection using this system?
So I think in terms of overall column selection, as I mentioned, there are a lot of different columns out there. We really talked about just, C18 reverse-based columns. That's, those are probably, the most common columns used in industry, but there are a lot of other options out there as well. The way the system is set up, you really, do have the flexibility to install columns from pretty much any vendor, as long as it has the standard 16th inch end fitting, which I'd say is probably 95+% of the column end fittings that are out there. So there's a lot of different options. In terms of dimensions, I would say that one thing I've noticed with this system is using maybe 3mm ID or 4.6mm ID columns is probably going to be the best bet, just in terms of the flow rate range that's available. But with the pressure limits that are here, you have a lot of column flexibility, because It works well with columns that have 400 bar limits, but also this system itself has a 10,000 psi limit, so you can push that 690 bar limit. So columns that are rated a little bit higher can be run a bit faster as well. But those, I think, are the big things. Are sticking to maybe a 3mm, 4.6mm ID format, but in terms of length or other phases, I think there's a lot of flexibility there.
All right, good to know. Thank you. And let's see, our next question. What, in your opinion, is the best advantage or advantages to moving to a miniLC or small system?
Well, I think that there are a lot of different sort of integrated small form factor LCs that are coming out. And that's part of the reason I've been excited about the system. It's a general area that I'm really interested in, and I think one thing is what is the general column scale. So some of the instruments that are coming out do have lower flow ranges. There might be some green analytical chemistry advantages there, but I think speaking about the miniLC in particular, I think the biggest advantage is, again, the small form factor that also is readily adaptable to a lot of existing instrumentation that's out there in terms of method transfer. So just as an example, a lot of companies put out very high pressure systems over the past 15 years or so, and they didn't see huge adoption. People stuck with more the standard HPLC range. And so you've actually seen in the most recent generation of instruments to come out, they've sort of been at intermediate pressures that fall exactly within the range that the miniLC hits. So I think that you're seeing that this is really the sweet spot of where modern instrument and column technology is sort of coming together. And that's the big thing, is if you're working on a method and you need to adapt it to some other site or want to bring it in-house or doing external testing, this is definitely a great price point for doing that. And I think the additional advantage is because it's analytical scale, you can easily adapt and transfer those methods. So that's a great question. Thank you.
Thank you. Alicia, do you have anything that you want to add?
Yeah, so sorry, I should have given you that option to say that. So yeah, I mean, Jim was right on the point of everything that he was talking about there, but just kind of from a very basic level, with a smaller system, you first gain, it's small, it's not very heavy, so it's movable, right? I wouldn't necessarily say portable. You're not going to bring it into the field, but you could easily say put it on a cart and move it from lab to lab. Jim even talked about the robustness and kind of saying, okay, different systems in different labs, but think about robustness of bringing one system to possibly different labs within a hallway or a building or so forth. I think Jim actually had an article not too long ago where he talked about point of need, point of care analysis, and that really makes that possible. And then also it's green, right? So it's small, it's only one standard plug, it's not using as much energy. Last week, Nick Snow talked about some of that as far as that small size opens up the ability that it is greener, and that is very applicable in today's world as well.
All right, thank you very much. At this point, I'm going to allow just a couple more minutes. So if you do have any further questions that have come up during this discussion, please feel free to add them now. I will take the opportunity to thank Dr. Jim Grenius as well as Dr. Alicia Stell for joining us today. This has been a great presentation. We hope that all of you have enjoyed it. And let me just leave the lines open for just a sec.
This concludes our presentation of utilizing compact integrated chromatographic instrumentation for pharmaceutical analysis. If you have further questions, please email organic.support@cem.com. Thank you for attending, and we look forward to seeing you at 3 p.m. for Dr. Emanuela Gianfrido's presentation on training the next generation of chromatographers using Lucidity Mini GC in the research and teaching laboratories. Goodbye.